6:30. Happy Friday. Welcome to another Small Grains for Brewing and Distilling virtual happy hour. This week we have Dr. Nicole Shriner from Michigan State University, and she'll be giving us an update on the MSU Fermented Beverages Analysis Lab that she is setting up and is very close to being ready and operational. So without further ado, cheers, Nicole, and tell us all about it.
Thank you. Welcome, everyone. Happy Friday. I will be honest, I just pulled this together really quickly. It was about a year ago-ish that I did a similar presentation, probably to a similar group. So some of the slides might look the same, but I have made some progress. So you just share that. Oh, will you help me, or you let me share?
Yes. All right, you're now co-host, and that's one thing I always forget. So
I always forget too. All right. You guys see that or no?
Yep.
Looks good.
Yep, you're good.
So if you don't know me, I am Nicole Shriner. I just want to give a background of me so you guys know kind of where I'm coming from, and if you ask me questions, whether I should know them or not. So I have a bachelor's and PhD in chemical engineering. My PhD was in cyclic distillation for energy conservation and spirit production. And basically that's just a different distillation method. So I got very savvy with the column that is in our lab right now. I've worked in industry at Red Cedar Spirits, which is a distillery in East Lansing. And I've also worked at two breweries, Harper's Brew Pub in East Lansing, as well as Old Nation Brewery in Williamston. After my PhD, I did a 6-month brewmaster program in Chicago and Munich. And when I came back, I started teaching all of the fermented beverage courses here, which led me to utilize a lab that had been left by my predecessor, Dr. Chris Berglund. He passed away unexpectedly, and the lab was kind of up for, anything to happen. So I kind of thought, why not try and utilize it to the best of its abilities and make it better. So that's currently what I'm doing. The goal of my lab is to support and enhance research and quality in the fermented beverage industry in Michigan. And I say in Michigan because I really want to focus on Michigan to begin with, but if that exceeds across the country, that's great. I want to be a local lab for industry members. The idea is that quality comes from consistency. Consistency comes from, being able to know if you're consistent or not, right? So collecting data. And if you can't collect data because you don't have the equipment, then you're kind of just repeating what you keep doing. But raw materials change, processing change, operators change. So being able to kind of quantify that and make sure you're keeping a consistent product is very valuable. So I want to be that middle person. If you want to know exactly what your IBU is, you want to know what your extract efficiency you've been getting from brewer to brewer. That's what I want to help with. So as well as for MSU researchers, a lot of them are sitting here in the meeting here, being able to maybe analyze and collect data at cost. So since we're in-house, we can't take margins. It's at whatever the internal federal rate is what we'd charge researchers. So that's just a kind of way to get more data with less money. So what I have, I'll show you a picture a little later, 3-tray copper batch distillation column that has a 120 liter capacity pot. We have a half barrel brew magic system. We have winemaking equipment that we use for the wine courses, but can definitely be used elsewhere. I have 4 7-gallon conical fermenters with heating and cooling control. Which are really fun. They look exactly like industry fermenters, but they're just lab size. Very fun. We also have filtering and bottling systems for wine, spirits, and beer. And newly added are a mash bath, finally. and friability meter. Those came from outside the country during COVID, so they took a very long time to get here. As far as analytical equipment, we have, I have 2 GCs in the lab here, an HPLC, spectrophotometer, Anton par densitometer, 1 liter bioreactors, which help to have a very strict control of a fermentation in regards to oxygen, pH, etc. Cryo freezer and centrifuge, so lots of microbiology equipment, and new large programmable laboratory oven for hopefully malting. In the future / coming from one of my grants I'm going to discuss in a little bit is a Headspace Sampler with Ecd Detector. And if you don't know what that means, that's okay. It's going to be used for diacetyl quantification and ethyl carbamate identification. So that is already in the works. I also have a wish list, so who doesn't? I had I don't know if Aaron's on the call yet or if he is at all, but I had a meeting with Thermo Scientific about a discrete gallery system for the enzymatic tests. They're amazing and so much more efficient with time and more consistent. So that's something in the future. I'd also like to look into sophisticated micro malting equipment, as well as a small scale mash tun so we can do everything in-house. Currently we're having another distillery mill and mash and distill into low wines, and to have complete control over the process we would need that to do in-house. I also am in the works of developing a sensory panel, just a list of people that throughout the country that are savvy in whatever we're talking about. So if it's hops, it's a hop panel. If it's rye, it's a specific rye panel. Just having a general consensus of people that know what they're talking about, know what they're smelling. And yes, you can train people, here in Michigan, and that's fine too. But I think reinventing the wheel here is not necessary. Just finding the people that know and having lists and contacts. So as far as what we offer now, so the James Decker and Christian, they operate a malting quality lab in the UP, and currently they do moisture, kernel plump, and thin germination tests, protein, pre-harvest sprout, and dawn. And the idea is we are going to collaborate to get all of the tests necessarily done. So that's so what I offer is going to be for grain malt and wort, viscosity, free amino nitrogen, fine and coarse extract, diastatic power, friability, color, clarity, filtration time, protein, both total and soluble, and alpha and beta amylase. And probably more what I'm more used to doing are fermentations or analyzing the final beverage. So for the color, the international bitterness units of beer, pH, ABV, and really looking at the fermentable sugars or the fermentable sugar array. A lot of mashing regimes can change, and that can change your overall amount of different sugars you have in your mash. So having an idea of what that looks like is very valuable. It takes HPLC to do that. Many breweries and distilleries do not have that. As well as alpha and beta acid concentrations for hops, ester concentrations, sulfur dioxide for wine. Something I'm really interested in doing is the absorbance fingerprint, which is a kind of newer idea. It's basically taking a sample and running a wavelength over spectrum over from low to high wavelength and getting like an absorbance fingerprint, and that would be what your product looks like. You don't necessarily know what all of the peaks mean, but it's kind of just a fingerprint of each spirit. So you can get a fingerprint of your product that you like, and then 6 months later, send another sample and see if those are similar. It doesn't necessarily tell you what's wrong, but it can give you an idea of a consistent product. So what are the practical uses in the beverage industry? If you're a distiller, you know what heads, hearts, and tails cuts are. Knowing what's in those fractions and seeing if those are consistent adds a lot of value. analyzing the mashing regime, like I just discussed, monitoring the brew house efficiency or extract and hop efficiency, recording your residual sugars. Also, there's producers getting into kombucha, so legally you have to be under a certain amount of ethanol for those, and non-alcoholic beers are getting more popular. So I can simply run that through the HPLC and tell you in 15-25 minutes if you're within the legal limit, which is very valuable. Analyzing barrel-aged or spontaneous fermented beers. There's a lot, sour beers are getting a lot more popular, and from my understanding through brewing school, a lot of people don't actually know what they're doing. They're just letting it happen and taking it out of the barrel when it tastes good. So have an idea of what acids are in that beer and how you're getting that the acetic or lactic or both, and what it looks like when the product is done would be great to know, and much easier to understand when the beer should come out of the barrel or what's even going on in it. Looking at different yeast strands, so maybe you're interested in using a different yeast, but you don't know exactly what that's going to do to your final product. That's something that I can help with as well, and just product development in general. So this is what mostly I wanted to talk about today, is the current projects that's happening. The next slide is going a little more in depth than this, but we're working on a rye variety project, which I think we talked about last week a little bit, looking at 6 or 7 different varieties of rye and understanding what those varieties would look like in a spirit, and if there is a difference, and what ones should be selected to grow specifically for spirits. The grain has been processed at a local distillery it's called Michigrain in Lansing, Michigan. They've processed it into what we call low wines, and now the low wines are in my lab ready to get distilled. The Hearts product will eventually go into barrels, and we're going to end up evaluating that over the next couple of years. I'm also working on a milk byproduct to vodka project. The Michigan Milk Producers Association have it's not just Michigan, it's the entire dairy industry that have lots of byproducts. That's why they're under a lot of heat, because their streams have very high BOD, biological oxygen demand, and they're trying to use these streams to add commodity. So what I'm doing is taking their stream and converting it into ethanol using various strands of yeast. Normal yeast cannot ferment lactose, but there are newer and engineered strains that can ferment into ethanol. And so subsequently making that into a vodka, it's not just about what one would make the best ethanol, it's what one would make the best tasting vodka. So that's a question that hasn't really been answered that I'm trying to help them answer. And the last thing that I'm currently working on is I just received a grant from the Michigan Craft Beverage Council again to do some research with bells, which I'm really excited about, on the production, reduction, and control of diacetyl in beers brewed with raspberries and fruits, specifically at bells. A main problem that's happening that has seen is normally diacetyl gets degraded over time, and that's kind of during your maturation stage. But they're finding in beers brewed with raspberries that doesn't happen. It doesn't come below technically threshold levels. Now it is passing sensory, I believe, because the raspberry is so overwhelming that you can't taste the diacetyl. But there's got to be some reason why that concentration won't come down. So that's what the research is in, and it's in the very beginning stages. We just received that, I think, a month and a half ago or so. So on the right, you can see those are the low wines from the 6 rye varieties. They came in totes and pallets, and the still behind that is where they're going to be finished. It's actually the perfect batch size, so I'll be able to fit all of the low wines into one run, and then I will distill them into cuts and analyze them further. On the left is literally the first data I've collected, and it's as of today. So on the left, you'll see the variety, and then I just kind of calculated the proof gallons of low wines per pound of grain. Not all of the mashes were the same amount of grain. but to average them out, I did the amount of proof gallons that were delivered to me divided by the pounds of grain that was given to the distillery. And I also matched that with the predicted spirit yield that was gathered. And it's not exactly the same, but the two varieties that were the highest in my lab were also the two varieties that were the highest here. So might be able to make a conclusion and say that they're equal, but it also has to do a lot with processing, and these numbers are all very close in general. So one thing that is really exciting is I've been starting to think about, you know, what product or what compounds are inherent in the grain that actually make it into the distillate. So I started to reach out to people that knew more than me about that. And I came across Keith Cadwalder, I'm sorry if I butchered that last name, at Illinois. And I just emailed him asking that question, like what he suggests about analyzing. And he was very interested in the research and offered to run the low lines on his olfactory system. He has a lot of experience working with rye and whiskeys, so he's going to do that. I just packaged the samples today, so that'll be exciting to hear back what he says. He's confident that if it would show up in the hearts, it'll definitely be in the low lines, so that'll be very cool to hear. Some future projects. Some MSU groups, Timothy Miles, he's in the small fruit and hop pathology program. He has contacted me about looking at centennial hops and seeing how a specific infection affects the quality of them. So essentially I'll be brewing some beer with fresh non-infected hops and the infected hops and kind of looking at the difference. So that's super exciting. And also some, there's a new grapevine that is resistant to some mildew, and we're going to look at making some wine out of it and seeing how kind of the quality of that. So that's pretty much my status and all of the info that I have for you guys. But if you want to ask any questions, I'm ready.
Awesome. Thank you very much. I just have a quick question about ethyl carbamate detection and just a clarification. So when we talk about glycosidic nitrile being present or not present in barley varieties, ethyl carbamate is the carcinogenic chemical that can be produced when reacting with copper from glycosidic nitrile. Correct me if I'm wrong on that, but yeah, it's pretty cool, I think, that we'll have that analytical capability to be able to run quality control on
yeah, so I was actually not very knowledgeable about that compound, and I did a little more further research, and I found that it's GC with the detectors that I do have and polar columns. So I have that. I need to get the standard and carefully evaluate that, but should be able to detect it in spirits. So probably not in, I guess we'd have to figure out the extraction method for that or the precursors for that, but in spirits themselves or final products in general.
Nice. Looks like we have a question from Tom. He wants to know, what was your mash bill for the various rye ferments and were they raw or malted?
100% raw.
So from Jeff,
portable DNA sequence.
Oh, there you
like
go.
A PCR. Is that what I've?
Oh, next gen, like an Illumina chip kind of setup.
I certainly haven't, but as I grow and find uses for more things, that could definitely be on the list. Always something to add.
So what, regarding the raspberry and diacetyl project, do you have any like ballpark suspicions about what's going on?
Yeah, it's pretty interesting the way that the yeast behave in a fermentation. I believe it has to do with the amino acids. So there's several different amino acids that get used throughout the fermentation by yeast. And there's some really fast absorbing ones and ones that not all get even used up. When there becomes a depletion in the precursor for diacetyl, it triggers that pathway to need it. So it will try and make that amino acid, and it offshoots into diacetyl. My thought is that if you add sugar or add, something to try and spike the fermentation back up, like a raspberry or any type of fruit or any type of sugar, it triggers that pathway into needing that amino acid. So it tries to synthesize it so your diacetyl gets built up. Another thing is we're also considering what is inherent in the raspberries themselves. Is there an enzyme that could possibly cleave sugars that were once unfermentable into fermentable, and now thus messing up the chain of command of the yeast back uptaking it? We could also be potentially hurting the membrane of the yeast cell by decreasing the pH with the raspberry. There's just a lot of different possibilities.
Yeah, interesting. So got a couple more questions popping in the chat here. So what yeast strain were used in the rye fermentations?
DAD, it's their whiskey yeast. I have it written down somewhere. It was the same yeast strain. I'll have to pull up what exactly it was. But all of the processing were supposed to be the same. The original gravity did vary a little bit, which is not ideal for consistent fermentation. But other than that, the process was told to remain exactly the same. And the spirit yield we got, it's kind of the same. I have all the samples that I kind of have been looking at today, and I do taste a difference. I can't quantify that necessarily, so we'll see what the analytics say, but I think there's a difference.
Yeah, I'm really excited to see what differences are there. We heard last week from Joachim, who unfortunately ran across a heavy geranium character that he didn't find very pleasing. Next question is from Michael.
Mm-hmm.
So what are the plans for commercial services at the lab, and what will be the capacity of the lab, turnaround time, and then maybe tours after COVID?
Yeah, great question. So commercial services, everything that I mentioned is offered to everyone. It's just going to be the rate is different. If an external person, there's a 26% increase from MSU plus whatever margin. Within MSU, there's nothing. So anything that I offer is also available to everyone else. Capacity of the lab, as far as what a test, or I don't know how to exactly answer that. It's hard to say.
Kind of the number of samples you're able to take in, and if you're let's say, I understand it's an early part of the plant, but I'm trying the lab. Sorry, I'm trying to understand. If we're urging people to work, and you're going to get 10 samples a day or 10 test, single test samples a day, or you're going to get 50 a day. I don't think the market's right there yet, but that seems like a lot of tests to do. So is it your goal is to have a couple come in a day, 5 come in a day, 5 a week come in a day, that kind of thing?
I think it's
what's your projection say?
Yeah, it's going to be more probably depending on the time of year. So, I mean, harvest is obviously in the summer, so I expect to be pretty busy in the summer. But the rest of the year, probably not going to be doing a lot. I think I'm primarily going to be focusing on these projects that come along, but as samples come in, processing them ASAP. Turnaround time, it depends on what it is. If it's something that you're testing a raw material, and if you want all of the malt analytics, that might take a couple of days. And also depending on what else I have, if I'm already working on someone else's samples. I also am going to hire an undergrad if we get more samples. So I will have work. I got approval for that. So kind of just going to go with the demand and try and, everything will be as quick as possible. That's all I can say for that. And as far as tours, as soon as the university opens up, I can have visitors. I'm under a very strict protocol. We had to get a lab return to safety lab plan approved all the way up to the president. So if I were to change that, when I add an undergrad, I have to get approval all the way up to there too. So it's pretty strict right now, but I imagine at least in the fall or the end of 2021, I should be able to have people in.
Excellent. There's always video tours too. I hear that.
Yeah, absolutely. Bells wants to do that. So that's maybe something I could do if we set something up. I don't know. I could probably show you right now, but it's a little messy.
Just have a comment in the chat too from Jeff again. Who worked at a phytochemistry lab at the University of Hawaii, Manoa. He worked focused on hard cider production. It's
Cool.
interesting. Yeah, big in our state. Reading it right now too.
Great contact.
Yeah. Stayed with hops DNA, which is really interesting as well.
Yeah.
Cool
I
stuff.
have a question. Nicole, would it be possible to share your slide again where you showed the preliminary rye data?
Yeah.
I'm just curious. Sorry, I'm like nerding out on these data.
It's not a lot. I don't really know if there's much to gather from it or not, but it's kind of cool to see at least preliminary stuff.
I think there's some interesting stuff going on here, both in terms, because what we didn't disclose to this group per se is that Danko and Bono came from the UP, right? And the rest of them came from KBS or around Hickory Corners. And if you dig deeper into the spirit yield numbers from Aaron, they actually track even closer than, so that's the average spirit yield for the 3 sites. But if you look at the specific sites, I think they track even closer than that. So I think your numbers are like spot on from what you found. Because generally the UP had lower spirit yield per Aaron's analysis than downstate for whatever reason this year. So
I'll get back with you about where each of them were grown and put that instead of the average, use the actual one. And yeah, we'll do that. Very cool.
Kind of related to that too, I'm looking at the rye variety trial, which I think is still in the chat from when Brooke dropped it in there first thing on the Zoom. But the 4VG, so the compound that we're using to track flavor characteristics in it in Chatham in the UP are consistently higher than at Hickory Corners. So there's like a flavor continuum going on there. We'll see. Interesting stuff.
I'm really excited to hear what Keith says about them. Because I can smell it. I know it's there. I know there's a difference. I just can't exactly quantify that.
Yeah, it was, it doesn't seem to be a thing with barley, but photoperiod sensitivity in rye was kind of a surprise to me when Joachim was talking about that and the differences between the height of his variety trials. So I know he said he was very tall, but his seemed to be like about the same height as around 1 meter as like barley. But rye is very much taller here. It's really, really tall. And that's apparently due to photoperiod sensitivity. Interesting stuff. And that I would guess is at play in Chatham versus Hickory Corners as well. Tom's got another question about milling. So how did you mill the raw grain, hammer versus roller, and what moisture level were they at? So I think Brooke could probably comment on moisture
Yeah,
levels.
I don't, we'd have to look at the data. You guys would know the moisture. And distilleries typically use a hammer mill. I didn't look at their mill specifically. Wait, did you have it milled and then shipped to them? I don't remember.
Yeah.
Did they mill it?
Yeah, Kevin at Emergent Malt milled it with a hammer mill,
Yeah, okay.
and to a, you know, as fine as he could get it. And then the moistures varied from, I don't know the exact, but anywhere from like 10 to up to close to 14%, I think, as harvested. And then we didn't dry them after that.
That depended on the site, Brooke, right? The KBS and the UPREC were around 10, and then that third site, the Gradium site, was at 13.
Well, these samples that Nicole has came from different fields, right? Because these were big blocks. Because in order to get enough volume, we had to grow big blocks. But for the variety trials that we sent to you, Aaron, we needed replication, and we couldn't do that in huge blocks. So,
Oh, sorry, I was confused. I thought you made the bigger ones by compositing.
no, we actually had like 1 acre fields that we harvested with the big combine, and then so in order to get that much volume. Yep. But yeah, the samples that came to your lab were composites of small plots, but replicates of small plots. So
We should have done the reps because we could publish it. In retrospect, I mean, it would have been expensive, but we don't have it. If we were to publish this now, we'd have to consider the sites as reps. We wouldn't have the G by E. We'd only have the G, the genotype interaction. If we had done reps within sites, we'd have the G by E.
yeah, I don't know if we still have the samples. I'll check to see if we still have them, or next year.
Next year, yeah, let's do it next year. Well, no, because if we did it both years, we'd have year. We'd have genotype and environment and year.
Yes, a true scientist. Yes.
I just mentioned it because Gary Spedding, not sure if you're familiar, you know with this, Nicole, right? They're launching a new professional society. I'm not sure the title. It's a Society of Distilling Science and Technology, and a new journal to go along with it. And this is sort of being led by Brian Christensen who publishes like a distilling magazine, Artisan Spirit, and Gary Spedding, who's a very well-known scientist and runs probably the best known analytical firm, Brewing, Distilling Analytical Services. And so that journal, they want to publish the first issue in the next quarter. They're seeking articles now on distilling science. This is a good one. I mean, the one we did just on the 4VG and the PSY with the genotypes, right? I was thinking about it. It's just that we don't have the genotype by environment interaction, but it would be statistical on genotypes if we just consider the environments as reps. So we would be able to show there's a significant difference among genotypes, both in spirit yield and 4VG, and the inverse relationship with protein. It's probably enough.
And you know, just thinking about this like plot scale to field scale in the same geographic area, but in different fields, and we're seeing similar spirit yield results.
That's impressive. Like now that you mention it, that we didn't even analyze the large plots, and it still correlates. Is very good. I mean, it's more, I think, a testament to the uniformity of how good you are at taking care of your plots.
Well, and the variety, the impact of the variety, right? If it's consistent between the plots and the field.
I think we know the variety that the rye, at least these parameters that we're measuring, things like protein kernel size, which essentially all of those effects we saw, right, on spirit yield, it was like driven by protein. It's very phenotypic, right? Like if you're a low protein variety with high yield, you're going to do that on almost every location. So we should maybe just write it up, and then you could do a follow-up story, a follow-up article on the big scale stuff, right? When Nicole gets all hers through. So we could do a small part one just on the, grain quality and a little bit about the predicted, what the lab is saying is going to happen. And then you can follow up later.
I like it.
So we have a question. Sounds like it's about ring testing. So what quality control procedures ensure the accuracy, precision, and repetition of tests? And are there other labs or groups to check calibration, et cetera? And you did mention using ASBC methods, but are you planning on participating in ring testing?
Yeah, so that's the first step I'm gonna take with that. I'm gonna be doing with Bell's. We're gonna be running the same test on the same materials and seeing how close we are, making sure we're good with that. And then as far as like the malting stuff, there's standard malt out there that is supposed to be a specific like this, and this. So that's what I'm gonna start with and make sure that I'm getting what I'm supposed to be getting. And then I actually was going to reach out to Aaron and see if he would work with me and say, I'll send you a sample, tell me what you get. And, that's a way to do it. But then there's also the ASBC certification program, which I potentially am going to do. That's down the road when I'm all going. But yeah, I'm going to double check and triple check, make sure I'm accurate.
Oh yeah, I think Pfeiffer is kind enough to send you a check sample of malt to run through your friabilimeter as well. At least they did for me.
Yeah,
That's what
I'm
you
so
get for
excited. It
$7,000.
just came the other day.
It's a super fun machine.
Yeah.
Got no other questions in the chat. This directly feeds into next week's topic too, actually, which is G by E interactions, so genotype by environment interactions, and we'll be hearing from Campbell Morrissey from Oregon State University. And then as a companion speaker to that, don't be mad at me, we'll have Rob Sarine, who will be talking about hop terroir in Michigan. So yeah, you already mentioned hops earlier, so you kind of eased me into that announcement. Thank you.
Hey, Ryan, I've got a question.
Sure, go ahead.
Hi, everybody. Nicole, thinking about the lab, and I believe that some of the equipment was new, some of it was used. What do you have in plans for maintenance, and what are the long-term maintenance needs? How are you guys going to cover the cost on that?
So thankfully, the rates, they include consumables. So since we can't, technically our rates can't include making, paying for that equipment again, because it comes from a federal grant. So we can't say, we need you to pay us for this because we didn't actually buy it. But it does cover consumables. And that's mainly the cost in maintenance. The other maintenance is just kind of me like cleaning and running the right fluids and taking care of columns and things like that. So a lot of it is just like me making sure that I'm maintaining the equipment. And then the others is replacing consumables as needed. And those are built into the rates. So that should be okay.
That's something we've really struggled with too. It's one thing to have a research lab where you work on projects, right? And you buy this equipment and you work with the people on projects. Maybe the money changes hands, maybe it doesn't. And then when stuff breaks down, you fix it. And if you have to wait to fix it, well, the project waits, right? What we've really found is once you hang out your shingle and people come to rely on you for QC and you have turnaround times and service standards, the stress and the hardship for your customers, your client base, if you're down, is really much worse, right? Because you have a service standard, and so they send in their sample and you say, oh, sorry, I can't because my whatever gallery analyzer is busted. So we had to invest in service contracts, and those are expensive, right? So we pay $3,000 a year, $3,500 a year, and make sure that means that someone will be at our facility within 48 hours to fix the bloody thing, because the clients can't wait. So yeah, it was a big different change in mindset from kind of a research center, which you kind of mostly are now, where something breaks, well, you fix it, to you can't, once you sort of, people come to rely on you, it was a big change for us in terms of like, well, we need to be able to ensure that we're not down, right? Because once the mass batch goes down, you're not doing anything, right? Samples just pile up. So yeah, we really have to think about the costs, and they're heavy duty, the maintenance contracts on these instruments.
Yeah, David told me to tell you hi from the gallery discrete guy.
David Blatz wants to sell you a gallery? Yeah, that's great.
Yeah.
And those are very good instruments. They really,
oh my gosh.
It's discrete, it's essentially a chemistry robot, right? And what it allows you to do is achieve the kind of precision that you need for quality control, right? If you try to do some of these diastatic power and alpha families, if you try to do those from the ASBC methods without automation, you never get that precision, right? And so It's a very flexible instrument. It's half the price of what the instruments used to cost, the continuous analyzers. But you should also ask David, since that's one of your planned and your planned purchases, to watch out for a demo for you. I bought a demo. It wasn't brand new instrument.
That's what I was gonna go searching around for a used one, because
don't buy a used one on the open market. Get him to sell you a certified refurbished. It'll come with a 1-year warranty, and it could be potentially like half the price. And don't get the Gallery Plus unless you really, really know you want to do the bitterness that way. Get the Gallery Regular.
Yeah, no, I already do the bitterness by manually, and it's fine.
Yeah, so that's like only 40 maybe-ish for the regular, and if you could find a demo with certified reused from him, he'll help you out.
Yeah,
tell him we're friends.
Yes, okay, I will.
He's a good guy. Oh, the other thing about that machine is they really want to sell you reagents. So Thermo makes most of their money on reagents, not on the instruments. It's kind of like Nintendo, right? It's all about the games. But you don't have to buy the reagents from them. So the newly published, they don't want you to know that, but the newly published ASBC methods that should be on the website soon for discrete have the instructions to make the reagents.
Wonderful. That's good to know. I was going to ask that, but then I knew their answer was going to be, you should buy it from us.
Yeah, their answer is going to be buy them from them, but you don't have to. And we have Hannah Turner at Montana State Malt Product to thank for that. She's the one who reverse engineered most of the reagents.
Wonderful. I'll have to thank her.
Very cool. And if I'm not mistaken, the Gallery would increase throughput and process flow and sampling, right?
Yeah, and decrease the cost for in-house.
Mm-hmm.
When it comes to malt testing, the pinch point in terms of throughput per day is the number of places in your mash bath. So I don't know what size you ordered.
I got a 12.
12 is great. Yeah, so 12 a day. That's your
There's
limit.
my throughput, Michael.
12 a day. Yeah, there you go. We've got the 20, so we're at 80 a week right now. We do about 80 malts a week. We mash 4 days out of 5. We try not to mash every day.
Very cool. So I don't know, just out of curiosity, what method are you looking at for IBU detection? Are you going to be using like UV vis, like spectrophotometry?
Yeah, that's what we learned in brewing school, and it's also one of the ASBC certified methods, and that's what I also teach to my brewing students too. So I've got a lot of experience with that, and it seems to be pretty repeatable. So
nice. When I was in the malt house, I was trying to figure out a way to justify bumping up to UV vis from regular vis, but yeah.
Yeah, it doubles the price of the instrument.
It does.
I got chemistry to buy that for me because we use a spectrometer for our wine assays, and I was like, we need a new one. It's broken, and so I just got the UV vis, and there we go.
Yeah, if you have an application that's that you can use it for, why not?
Yeah.
Cool. Well, any more questions from the attendees? Anything we want to talk about?
I think it was
I have
a good
another quick
segue.
question.
Oh, I'm sorry, Janelle. Sorry about that.
Sure.
Oh, thanks, Michael. You may have said this, but I was just wondering if you are currently doing any analysis for commercial purposes, or is that not happening yet?
So as long as there's none of my bosses on the line, I have been, but I haven't got the rates approved yet. So I haven't charged anyone, but I have helped. So someone at Doemens, they're making a non-alcoholic, and he's doing he's doing like a reverse filter system. I guess I've never knew that method, but he wanted to know the alcohol percentage if he was low enough, and he wasn't. So I ran that. It took 15 minutes for me to tell him that he was 0.5% over on his ABV for his non-alcoholic beer, which was pretty cool. I've also ran for, what was the other thing I ran? I do a lot of wine samples for my colleague for lactic, malic, and alcohol. That's super quick, and it's a pain in the butt to do all of that yourself. So running that through the HPLC, I get data that would take them days to get. So done that. But when my rates are approved, they'll go on our website, which I guess we could have talked about too. All of the analysis will be show on the website, so you can understand what's offered and how much each thing is. So once that's happened, my plan is to kind of reach out a little bit more to like the Master Brewers Association, reach out my contacts in the wine and distilling industry and tell them, you know, I'm up and running and these are the things I think I could help you with. So getting very close to being more commercial, but got to get those rates approved first.
How did you develop your costing model? Like when you were doing that, did you look at some of the other comparative prices? I hope you didn't undercut the market really bad, because we're already given this shit, but...
I can't do that. So I used Gary's website, and my margins, I just made them as big as they needed to be to match basically Gary's. They might be a little bit less than his in some aspects, they were more. So that's one thing that I really didn't want to do. And I feel like there's got to be an illegal aspect of that, like using federal bought equipment to undercut privately owned businesses. I don't know. But we, yeah, the prices in-house, they're dirt cheap, but what industry members will see, I'm not undercutting. And I mean, it's consumables, cost, reagents, and then time. And the time thing was actually the hardest, because I'd potentially like to hire like a graduate or undergraduate student to do these, because they're not, you know, rocket science, and I want to focus on the research. But right now it's me, so kind of finding a happy medium rate that works for not only me but also a grad student. So.
Michael, do you have another question?
Yeah, I was just gonna, I was quite inspired by last week's topic, and I think the data and what, what the extension of the data is. And I think that where I might have missed some points or missed some comments was the scale up. We asked about the rye acreage. We asked about this, elemental decision, whether this is raw or malted. And then there was, everyone should know that I'm not technical. There was quite a bit of discussion, I think, about flavor and the chemicals that might be brought in that are potentially making that flavor. Sorry, and I thought that was a great discussion. But I think the data here is really hypercritical to those decisions. So again, it's going to be the agronomic and malt house and brew house distillery, and also that whole sensory conversation we had. So I just want to say I'm super excited. I guess I wouldn't say it any other way that Michigan State University, the reason those samples were so good is because Michigan State University research is world class. And
Thanks,
I hope we could, yeah, it's truth. And I just wanted to keep going to get to a commercialization point. And I want us to focus on demonstrating the scale up as well as the sensory thing, which I think is still we're just at the scratching the surface on rye sensory. And you'd think as someone just been part of the industry for several years that maybe that's been done, or that there are known elements, and maybe there is, but we need to find that or get data that tells us. So I'm super excited about the rye thing, and I think that should be a big focus, and I think it will go hand in hand with the barley research as well.
Yeah, absolutely. And related to that topic, and web presence as well, we're in the process of making our malting barley research team, our web presence more in line with what we're doing with rye, with oats, with other things, in concert with Nicole. I dropped it in the chat. I'm basically reiterating what I just typed. But yeah, we're in the process of making our web presence more indicative of the work that we're doing, and that it's not just malting barley. So yeah, we're pretty excited about that, and we're also learning a lot of new things about how websites work. All right. So any other questions or anything anybody wants to discuss? Doesn't have to be about rye. Could bring the hop thing up again. I'm okay with that. No, okay.
Aaron.
It's not okay with that.
I'm a grain guy. We're grain guys.
It's in
This
the
is a
title of
grain
this session.
Happy hour. Hops get enough attention.
That's true. I have a hop tattoo to prove it.
Seriously, now why don't you have a barley tattoo?
It's in the works.
It looks like a mole. It just looks like a mole if you just do that. So, no, there you
Yeast
go.
tattoo. That's funny.
Yeast tattoo? Just a dot.
Oh, yeah, maybe it's budding.
Yeah, it's budding. There's two.
Yeah. Well, cool. Well, thanks everyone for being on the call today. It's lovely to see your faces every Friday. I always enjoy it. Again, to reiterate, next week we'll have Campbell Morrissey from Oregon State University presenting about barley terroir research that is ongoing there, highlighting that genotype by environment interaction and how that ends up manifesting in your glass and what you taste. And then to bring that into a regional form of relevance, it will be Rob Sarine talking about hop terroir. I can't stop talking about hops today. But I think that really puts it into perspective. There's a lot of deep research on hops in Michigan as well. So yeah, it'll be an interesting session and something a little bit different than we've been talking about already. So thanks again for everyone being here, and I'll see you all again next week. And cheers.
Thanks, everyone.
Thank-
Good to see you.
Thanks for hosting.
Cheers.
Have a good weekend.
Bye, everyone.
See you, everybody.